In this presentation, we will look at venom and the antidotes that are currently in place for them. We are looking in particular at the venom of the black mamba snake, the most deadly snake on the planet. Its venom can kill a person in seconds if not treated properly, and its venom is one of the most neurologically damaging substances in the world. The reason why this venom is interesting chemically is because venom is being researched currently to see if its effects can be used in a more beneficial way to help fight disease. Neurotoxins could possibly be used to treat brain injuries, strokes, or even Alzheimer's disease. First, we will go through the chemical makeup of venom. Then we will review how each of the components has an effect on the body and what chemical processes allow this to happen. Then we will look at treatment for black mamba bites and what chemical processes break down the effect venom has. Lastly, what the chemical makeup of the antivenom is. Venom is mainly made up of all proteins. These proteins are divided into different subcategories that include neurotoxic peptides, which includes dendrotoxins, then other toxins known as calciseptin and membalgins for black mamba specifically. There are also proteases, hyaluronidase, phosphatase, and phospholipase. Each of these work chemically to have different effects on the body, ranging from breaking down neurological communication to breaking the cell walls to stop cells from functioning properly. Before we look into that, first we will go over the chemical makeup of a protein itself. Proteins are made up of amino acids, which is what is pictured here. Amino acids are made up of an amino group, a carboxylic acid group, and a side chain that is composed of carbons, oxygens, and hydrogens. Amino acids bond together to make a protein through what is known as a peptide bond, which are covalent bonds. The way they bond is the carboxyl group of one amino acid and the amino group of another amino acid will bond together and release an H2O molecule. This is known as a dehydration reaction because water is a byproduct and is being released as a result of the amino acids bonding together. This then forms the polypeptide chains, which are simply two or more amino acids bonded together. Finally, to go from a polypeptide chain to a protein, there are more chemical processes involved. Polypeptides are made from multiple amino acids linked together. Likewise, proteins are made from multiple polypeptides bonded together. The polypeptides together is what is known as the primary structure of a protein. Then the secondary and further structures come from more intermolecular bonding. The secondary structure is created from hydrogen bonding between amino groups and carboxyl groups in the primary structure, and these then fold the protein to give it its shape. The first protein we will look at are what is known as dendrotoxins. Dendrotoxins are proteins that cause neurological damage in the body when venom is injected. Specifically, dendrotoxins block potassium channels in neurons, which is one of the ways our neurons signal other neurons. Dendrotoxins have a positive charge and a neutral pH, and potassium channels are regulated by positive ions of K+ and sodium+. These two regulate the channels. However, dendrotoxins, because they are overall positive, they can mess with the regulation of these channels and break down the neurological signaling. The positive residues in the amino acids lysine and arginine, which are very present in this protein, are what help it bind to the neurons, as well as overload the neurons with positive charge and stop the potassium channels from working properly. The NH2 groups on these two amino acids are what make the overall positive charge, because they are positively charged ions that affect the neurons that the toxins attach to. This is incredibly damaging because it shuts down neurological signaling, which in turn results in the breakdown of neurological function. The next is known as calciseptin. These are another type of toxin that break down L-type calcium channels and inhibit them. Calcium channels are used for regulation of the heart and cardiac function. The calcium channels are used for the regulation of electricity in the heart, and the charged calcium is what does this. The way calciseptin works is they reduce the electrical conduction to the heart, which then decreases the force of contraction of the muscle cells, which finally dilates the arteries. This all, in the concentrated dosage of the calciseptin in the venom of a black mamba, shuts down smooth muscle function and especially cardiac function. Calciseptin has heavy amounts of the amino acid proline in its composition, and the proline helps the protein bind to the calcium channel receptors and block activity through hydrogen bonding at the NH2 sites. Lastly, there are membalgins, which unfortunately the least is known about. These components of the venom are responsible for breaking down acid-sensing ion channels in neurons. Acid-sensing ion channels are in general responsible for the regulation of pain in people and how they perceive pain. The membalgins bind to ion channels and block the flow of ions into them, thus blocking signaling and the ability to feel pain. These are a numbing agent that is in the venom and the last neurological component of the venom. The membalgins bind to the acid-sensing ion channels through hydrogen bonding with three main amino acids, lysine, arginine, and histidine. These then trap the chemicals in a closed conformation, so ions can no longer penetrate the membrane and form signals. Lastly, there are other components of black mamba venom, such as phospholipase. Phospholipase is a protein enzyme that breaks down the cell membrane. It destroys ester bonds through hydrolysing them, and ester bonds are important in the connection of the cell membrane. Hydrolysing refers to using water to break chemical bonds. This then results in the cellular contents being spewed out and decreasing the cell function when the phospholipase is active. Then there are proteases, which break up proteins and blood hemoglobin through hydrolysing, just like the phospholipase. The proteins, they hydrolyse the proteins and break the peptide bonds there, which destroys the structure of the protein. There are also phosphatases, which fragment ATP and take away the basic cellular currency. It removes phosphate groups from the ATP and then breaks down their function. Lastly, there is hyaluronidase, which breaks up extracellular components between cells, allowing for a more rapid spread of the venom, just as the phospholipase broke it down as well. Each of these result in the rapid spread of the venom, so the neurotoxins mentioned above can have a more damaging effect on the body.
Let's discuss the standard treatments for black mamba bites, which generally match those of most venomous snakes. There are two general steps that need to be followed after a bite occurs. The first thing that one should do is bandage or immobilize the bitten limb with crepe bandages and a splint. This prevents excess bleeding from occurring and helps to limit the spread of the venom in the body. Next, antivenom from the South African Institute for Medical Research, or SAMR polyvalent antivenom, should be administered. Now let's talk about how the antivenoms are made. First, venom is extracted and collected from snake fangs. This venom is then used to create vaccinations, which are administered to donor animals such as horses or sheep. These animals then produce powerful antibodies that are able to bind the venom from the vaccine. Antibodies are essentially proteins that look to fight and neutralize any antigens or toxins. The original vaccine is not administered to humans because we are unable to produce antibodies powerful enough to neutralize venom as the donor animals can. The antibodies are harvested and purified from plasma that was also produced by the animals. These purified antibodies then go on to become the main components of classic snake antivenoms, such as the SAMR polyvalent antivenom used primarily for mamba bites. There are three types of antibodies that can be used for snake antivenoms. The first and the most common is the immunoglobulin G antibody. The next types are the fragment antigen binding antibody and the secondary fragment antigen binding antibody. As the name implies, these two antibodies are fragments of the immunoglobulin G antibody. The secondary fragment antibody is about double the size of the regular fragment antibody. Now on to the specifics of the chemical composition of the antibodies themselves. A full immunoglobulin G antibody is made of two polypeptide chains. As discussed before, polypeptide chains are basically strands of multiple amino acids. Of the four polypeptide chains, there are two heavy chains and two light chains, where a heavy chain is about double the molecular weight of a light chain. Each heavy chain has one variable domain marked as VH and three constant domains marked as CH1, CH2, and CH3. Each light chain has one variable domain, marked as VL, and one constant domain, CL. The heavy and light chains are held together by several disulfide bonds. The variable domains are the areas that are considered the antigen binding sites, meaning the part where the antigen or venom molecule is attracted to and neutralized. The secondary fragment antibody consists of the branches above the hinge region of the original immunoglobulin G antibody, while the original fragment antibody is just one of those branches. Now let's discuss how the antibodies neutralize the venom. When an antibody binds to an antigen, it is able to neutralize its poisonous properties by changing its chemical composition and switching around elements. The antibodies work with the body's B cells to trap and neutralize any toxins that are found. The antigens are able to bind to the antibodies due to both of their powerful van der Waals forces, dipole-dipole interactions, and London dispersion forces. There are four main processes that happen when antibodies combine with antigens: precipitation, agglutination, phagocytosis, and hemolysis. Precipitation is simply the reaction of the antigens with the antibodies to form a precipitate, thereby neutralizing the toxicity of the venom. Agglutination, phagocytosis, and hemolysis are processes that essentially lead to the absorption of any antigens or toxins. In conclusion, black mamba venom and snake venom in general have damaging effects on the human body due to their dangerous chemical composition. However, good quality antibodies can effectively neutralize them, rendering them essentially harmless due to its variable polypeptide chains. However, there are no guarantees. It is imperative that people in communities with a high prevalence for toxic snakes, such as black mambas, take the time to recognize the chemistry behind their venom and respective antivenoms so that more effective action can be taken towards the treatment process.